I hope you are all doing well, and I want to go over the next phylum. It's a one-time dealio here. We're just going to talk about it once, and then I will also be posting a little bit of video support with that, and then I think maybe do a short quiz on that. And so speaking of quizzes, if you haven't already taken the Annelida arthropod quiz, go ahead and do that first. And if you took it and you didn't like the results, retake it and get a higher score. That's fine too. So I'm going to go ahead and share the screen here so that you can see my notes that I'm happy that I've kept from a previous year, because I can just go over this real quick. So this is Phylum Mollusca. So in the video with the arthropods, with the mantis shrimp attacking a snail for food, it talked about the snail being able to punch and destroy, to shatter the snail's shell so that it could eat it. And thanks to those jointed legs, boom, boom, arthropods can really pack a punch. They can swing. When they hit, there's a lot of force behind it. So the skeletal, the exoskeleton system, the jointed limbs and the muscles attached to those jointed limbs allow them to do that. And it's same with us, with our internal skeleton and our muscles attaching to our jointed skeleton. That is what enables us to have force in movement, whether we're punching or we're running. And so we can get some high impact stuff done. So Phylum Mollusca, now these are mushy creatures without jointed legs, mushy, mushy. And we're going to talk about those. So when I brought up snails, the snail question I asked from your mantis shrimp video was, how would that snail feel being punched? And that's a really higher end question hidden behind a simple thought. You know, oh, how did that feel? And it was fun seeing some of your comments about, my hair's gonna bother me. Oh, it would feel scared, it would feel terrified, you know, and some would say, well, it would feel pain. And at what point can we assess the feelings of something that's a very simple animal? So arthropods have brains, and in fact they have very simple brains, and yet their simple brains allow them to do magnificent things together. So I'm skipping a little, sorry, because you also watched the video on swarm theory. So simple brained things can actually, as a collective, manage some very complex group behaviors. So that's cool. When we're talking about braininess, it's like, well, one, how much brain is there? And then two, what can they do with that? We don't always, we can't know the answer to that, because any more than I can really know how you're thinking and feeling. How could we know how a snail is thinking and feeling? It's a lot easier for me to project how you would feel based on how I feel, and that we would share very similar responses to things, or understand, you know, what an emotion like anger or but that gets trickier when we're going across to other animals. So one thing we do is we compare brain size, and then we also compare brain wiring, like what do the parts of the brain that these animals have, what parts, what do they control, and how much can we relate that to our own brains? Or in other words, where can we find parts that are homologous? So I hope my cursor shows up on this. I think it does. So I'm circling the word homologous. If you remember, that's structures from the same evolutionary genetic origin. So that was a lot of setup. Let me see if I can run through this fairly quickly. So we've got 3 classes of mollusks, and they're neat to talk about because they're showing a range from very simple into medium complex to way crazy complex. And our 3 classes we're going to talk about are the bivalves. Those are like clams and mussels, and so these double-shelled animals that open up with a hinged side, and they've got muscular foot type things. It's a really big muscle that can protrude out from the inside of the shell, and then the animal walks around like that, you know, with the muscle sticking out, uses that foot to motor around. Now this muscle, if we look at Hox genes and development, it's very easy to see that this is a homologous structure to the muscular foot on a snail, which is the part of the muscular bottom part of the snail that comes, that pushes out of the shell that it moves around with. Okay? And another thing to point out is the calcium carbonate shell is homologous to the calcium carbonate shell up here. So those are two of our classes, Bivalvia and Gastropoda. And then our third class is Cephalopoda. So let me back up for a moment. Not much brain going on here. And in green, it says several paired ganglia. So pairs of ganglia, and I don't even know how they're kind of lined up inside of this, but so there are along the nerve system, there are just points where there are condensed areas of nerve cells, and that helps with the organization of this animal. And then here we have a cephalization of the ganglia, so we can say that snails have a brain. And if you look up at the top of the turrets where the eyeballs hang out, remember eyes are part of the nervous system. They're actually extensions of the brain. We did that when we did the flatworms, where we did the cutouts with the, remember the paper cutouts, and you guys added organ systems. So same thing going here, and there's the nerve cord going deeper into the animal. And I bet y'all know that hey, octopuses have big brains, they're super smart, and you've seen footage of them opening jars and being able to do very clever things. And so scientists are real interested in how brainy an octopus is. Now their brain pattern is actually set up not super aligned with the way vertebrate brains are. So it's like somewhere the fork in the road, the pathway to be an octopus, their brain organization is different than ours. And so it's hard for us to understand how it is they're able to think as well as they can. Super brainy seem to be able to think on higher level, and we don't know questions like, does an octopus feel fear and anger and you know, things would impose on, but boy, they can do some complex things. So bivalves, not too brainy. Gastropoda, kind of brainy. Octopuses and squid, way brainy. So one more thing I want to point out is the second word up here is vestigial. The word vestigial is meaning a structure that is a small genetic remnant of a bigger structure. So vestigial organs are, we can locate them as being homologous or being structures that are of the same genetic origins of another animal. And but along with that, they've changed purpose quite a bit, and they may have changed purposes in a way. Vestigial really is talking about it kind of shrinking or becoming less important, or a smaller portion of the animal. And we can find an example of a vestigial structure in the octopus. And it's not the brain. The brain's gotten brainier. It is growing in size and become, you know, the derived trait here is towards more braininess. The vestigial structure is looking for the calcium carbonate in an octopus, which isn't a shelled organism. A squid doesn't have shell. But look at this, calcium carbonate shows up where? On the beak. The beak is considered to be a vestigial structure to the shells of its related family members here within the phylum, or phylum members within the Phylum Mollusca. And so that's pretty nifty. And so vestigial structure is a word that I'd like you to try to take in and think about. Vestigial structures in, oh, sorry, school bell. I'm close to done, by the way. I'm close to done. I'm leaning to the side because that's where the little picture of me is. I got to remember to look at the camera. It's so confusing. So, vestigial structures in snakes, because I do love some snakes. Boa constrictors. I wish I had a male because I could show it to you better. I'm going to put this on pause, see if I can get it to show up on my little female. Hold on. All right, stop, share. Okay, so here's a boa constrictor booty. I hope she does not poo because she's making gurgling noises happening. an interesting video, and she wants the heck out because this was abrupt for her. All right, so on her tail, oh girl, never put a snake around your neck. That's a bad procedure. I don't want to do bad procedure. Calm down, Snakey. What I'm trying to show you when she comes down, surely it'll happen, is right at her cloaca, which is the where everything comes out, whether she's laying eggs or pooing or releasing urates, which are kind of like solid pee. It's the white chalky stuff you see from birds and snakies. So right there is a vestigial hip. And I'll show you the other side, but it looks the same, and this is complex enough. On the males, they're larger, because on the males, they're actually using them like little grabbers to hold on to the females during reproduction. And so just like, if you get my finger under, it's so small, and she's trying to knock everything off my desk. Ooh, that's pretty good. Right there, like I can feel it, it's sticking out. You just that scale right there, that leads into hip bones on the inside of the animal. Don't break the mug, snake. So she has a vestigial structure to a hip bone. Why would that be? Because ancestors had legs. And then we don't see this in snakes that we would say are more derived, which are the non-constricting snakes. So boas and pythons have those vestigial structures, but the majority of snakes don't have those vestigial structures. They're more derived, and they've lost the hip bones. Okay, so now I'm going to put it on pause and put her up. Okay, so just to recap, before I let you go here, Phylum Mollusca has these three classes, which are bivalves, gastropoda, and cephalopoda. Those are the clams and mussels, the snails, and the octopi and squid. And then also vocabulary terms are looking at homologous structures, or homology is the study of organs or structures between taxa that have the same genetic origins. And how are we checking those? We're looking at things that originate from the same Hox genes, same sets of Hox genes. And then the other term is a vestige, or a vestigial structure is a structure that is an evolutionary remnant of a previous structure. So how could we check that? Is to see where's that calcium carbonate being coded for along the genetic code, and finding the same Hox genes. Now, early on in the study of these of organisms, we didn't have Hox genes to rely on. We couldn't look at the DNA, so it's kind of fun to go back. And when you say, well, we've been talking about, oh, look, here's this calcium carbonate showing up, I bet that's a vestigial structure, you know, and that shows, kind of helps see the relatedness between these rather different looking organisms. But now we can look towards Hox genes, go, yep, sure enough, that's where that calcium carbonate is being coded for. That's where it's being built from. And so that's an interesting piece to a puzzle along this tail. And I think I've said pretty much what is on my screen, but you can always pause and check my work, see if there's anything else, and ask me questions like, oh, the vestigial structures on a fly. Additional resources. I don't know, so many things we could do, but we want to keep it simple, don't we? All righty, so have yourselves a great rest of your day, and I'll see you next time. Bye-bye.